Your lunch. I think most of us have made it back to the meeting room. So in view of time, I would like to start with the afternoon session. And first I would like to invite Diane Sanders to the stage to talk about by far the largest genomes that we've seen so far, the rust genomes. So Diane, the floor is yours.
There we go. Are we on now? Okay. So I'm a group leader at the John Innes Centre in Norwich in the UK, and I'm really interested in how we can actually harness the power of genomics to inform disease management, with the ultimate goal of trying to impact the farmers directly in the field, such as the smallholder farmers that are shown here. And so in my lab, we work on really emerging and re-emerging plant pathogens. And we're really interested in these emerging and re-emerging plant pathogens because over the past couple of decades, we've seen an increasing number of emerging diseases. And now these oomycete and fungal pathogens, they're posing a significant threat to animal health and food security. But also in those past couple of decades, we've started to really feel the impacts also of climate change driving our pathogens into new territories. And so it's been predicted that fungal pathogens alone, well, they're moving on average around 7 kilometers northwards every year, and that then drives these pathogens into new regions, making them very unpredictable. And we've already heard this morning about historically one of the most notorious disease epidemics that occurred, the Irish potato famine. And the reason that the oomycete pathogen that caused this disease was very successful was the fact that it's very adaptable and it's rapidly able to overcome plant resistance. And so there are a number of characteristics that really make these filamentous plant pathogens highly successful. Another characteristic is the fact that they exist in very large population sizes. And so you can see here on this piece of farm equipment, and what looks like mud caked on this farm equipment is actually the spores of this corn rust pathogen. And so they exist in vast population sizes. And they mix this asexual reproduction with sexual reproduction. So the asexual part allows them to generate lots of clones of themselves very, very rapidly. And then they mix that with sexual reproduction to generate new genetic variants, which are then able to potentially overcome plant resistance. And also the spores that they produce, these plant pathogens, they are also remarkably resilient. And so the wheat yellow rust pathogen, which is one of my favorite pathogens, it produces these beautiful asexual uridinospores. And those asexual uridinospores can travel vast distances, even between continents, to spread this pathogen around the globe. However, of course, these spores don't just have the potential to blow on the wind in order to move between different plant hosts. They also have the potential for catching on your clothing, and you can also help to move these pathogens around the world. And I think this one illustration here really sums up the potential for people to facilitate the movement of plant pathogens around the globe, as this shows the number of people that are using air transport in just one single day. And so as everyone continues to vacation in more and more exotic locations, that helps to bring these pathogens out of those regions and move them into new countries. And so as we are all aware at this particular meeting, it's absolutely essential that we have very well connected international surveillance networks to keep pace with these ever-evolving threats that we face. And so in my lab, we're particularly interested in a group of plant pathogens called the wheat rust pathogens. They're also known as serial killers. And they consist of these 3 different fungal plant pathogens: stem rust, leaf rust, and yellow rust. And the reason that, of course, we care so much about these pathogens is because of the devastation that they cause. So collectively, the 3 of these pathogens, they cost us globally over $3 billion every year to control them. They occur in all the major wheat-growing areas around the world. However, although they are economically extremely important to study, in practice, they're particularly difficult to work with. And I think one of the major reasons that they're very difficult to work with is that they're all obligate biotrophs. And so this means that you can't culture them in the absence of their plant host. And so you can't grow them in a petri dish, as we heard earlier about bacteria, we have to grow them at all times on their plant host. And so that just makes them very, very difficult to work with. And that, of course, transcribes to the fact that these particular pathogens are probably understudied when you compare them to other filamentous plant pathogens which have also got significant impact. And however, the genomic area has then provided new tools and technologies which have opened up research into these plant pathogens and provided new opportunities for us to better understand how these pathogens cause disease. And so in my lab, we've been trying to see how we can harness the power of genomics to try to improve pathogen surveillance with a focus around these complex fungal pathogens. And so one of the techniques that we developed around 5 years ago now in my lab was this field pathogenomics methodology. And so how this method works is we work either ourselves or with collaborators to go out into the field and identify infected leaf tissue. And so for our yellow rust pathogen, my favorite pathogen, this would be an infected wheat leaf. And then we take that back to the lab and we do transcriptome sequencing. This allows us to get sequences from all of the genes that are expressed in both the pathogen and the host at the same time from that infected wheat leaf, and also to understand the interaction between the plant and the pathogen directly in its natural environment. And so you can use the data then that we generate using this method to genotype the plant and its host, the pathogen and its host, so you can genotype which strain of the pathogen you have, and also understand maybe which wheat variety you have in that sample, and then understand how they're interacting with each other. And so we've used this methodology to address a whole array of different questions. So for instance, we used this back in 2016 in response to the wheat blast outbreak in Bangladesh. We worked closely in a project that was led by the Sainsbury Laboratory in Norwich, where we got samples from collaborators in Bangladesh where they were seeing this new disease in the field. And before 2016, it was thought this wheat blast pathogen was largely confined to South America, but then they were seeing what looked like the symptoms of this wheat blast pathogen in Bangladesh. So we got those samples from Bangladesh, sequenced them using this method, and then we did some comparative genomics, and through that analysis, we were able to determine that yes, indeed, it was wheat blast causing the disease that was being seen in Bangladesh, but also the likely route of entry due to the comparative analysis that we did with samples from other countries. And so this then provided not just the information that allowed us to confirm the disease, but also the potential route of entry into the country. And all of that high-resolution data and analysis was carried out within just 6 weeks of getting those samples from the field, and that information immediately then went out to the community. And also we used this method to track the dispersal of individual strains of pathogens. So for instance, my favorite pathogen, wheat yellow rust, I've tracked the strains of this pathogen all around the globe. And then we're also using this data to monitor mutations in important genes in the pathogen. So for instance, of course, one group that are very important are those that encode proteins which are targets of fungicides. So we're always trying to monitor those for any mutations that could mean that the corresponding proteins are no longer able to interact with those fungicides and thereby make the pathogen insensitive to the fungicide application. And then another area that we've been also working on is how to link these dispersal patterns to climatic models more recently, because this allows us to then get ahead of predicting future threats to our country. And one really important future threat currently in Western Europe is the re-establishment of the wheat stem rust pathogen. And so wheat stem rust seemed to have been largely eradicated a few decades ago from Western Europe, but over the past 5 or 6 years, it seems to be potentially re-emerging in Western Europe. And so we're looking at linking the analysis that we're doing with climatic modeling to look at the risk of that pathogen re-establishing. Okay, but today I just wanted to also mention how this kind of high-resolution genotype techniques can also be utilized to study the host side of the interaction, and how the data that we generate on the host side of the interaction can also help us to inform disease management. And so when you study the host side of the interaction in these infected field samples, you get data on how the pathogen is actually manipulating the host itself. And so if you can understand how the pathogen is manipulating the host, maybe you can identify genes that the pathogen is turning on in the plant host, which are essential for that pathogen to cause disease. And then if you were to disrupt the function of those genes, you would imagine that the pathogen would not be able to cause disease. And so those are the genes that we're trying to identify in these data sets. And so to do that, what we've done is we took samples from the field of varieties of wheat which are pretty resistant to our yellow rust pathogen, and then other varieties which are very susceptible to our yellow rust pathogen, and then we're looking for genes which are specifically turned on in the wheat variety that is very, very susceptible. And so we use our field pathogenomics strategy, we do our transcriptome sequencing, and then look for those genes that we can link to susceptibility. And then, of course, this is bioinformatic analysis, so we come up with a list of candidates, but then you have to take those forward and confirm whether or not they do have a role in disease susceptibility. So to do that, we disrupt the function of those genes and then test them in pathology tests. And so the ultimate goal is to provide new genes and markers that can be integrated into breeding material to improve disease resistance. And so today I just wanted to illustrate one of the many genes that we've been working on. And this is a gene which encodes a protein called Bcat. The function of this protein is not really important for today, but I wanted to show what happened when we actually disrupted the function of this gene in this wheat variety. So, apologies. So you can see here, this is the wild type plant, which is heavily infected by our yellow rust pathogen. All these spots here are all pustules of our yellow rust pathogen. And then we took this variety and we disrupted the function of this Bcat gene, which we'd hypothesized was important for the pathogen to cause disease. And then we infected those plants, and what we found was we had pretty much green leaves. And so the pathogen was completely unable to cause disease on these plants. And so this tells us that this Bcat gene is really essential for the pathogen to cause disease. And so then we can take that gene forward, and our breeders can either screen for disruption in any varieties that they might have, or indeed they can disrupt the gene in lines and then use those in their breeding programs. And that allows us to then provide them with a completely new source of resistance that could be used for disease management. Okay, so you can really see how powerful this field pathogenomics method is for gaining information on both the pathogen side and the host side of the interaction, and how you can use both of these sides to inform disease management. But for me, this didn't really go far enough, because I think this is a really great tool, and I'd like to see it in the hands of everyone. And so we wondered if we could actually take this methodology and convert it in a way that it would be more accessible to particularly resource-poor areas, so resource-poor countries, but also labs that just don't have access to really high-level sequencing facilities or high-level computational expertise. And this would then enhance the disease diagnostics capabilities, either in those countries or indeed in those labs. And so to address this, we decided to focus on one particular country to trial this out. And the reason that we focus on this particular country is because we chose Ethiopia, as Ethiopia is the largest wheat producer in sub-Saharan Africa. It also acts as a gateway for yellow rust strains moving from sexual populations in the near Himalayas region into East Africa. So it's kind of the gateway where all of these new genetic variants of the pathogen move into East Africa. And so it's a very important country. But also, Ethiopia suffered the worst yellow rust outbreak in recent history back in 2010. During this outbreak, it affected over 600,000 hectares of wheat. That equates to over a third of the country's wheat production area. It cost them over $250 million in losses. And so this was an absolutely catastrophic epidemic that occurred in the country. And of course, Ethiopia, being a developing country, didn't have much resilience. And so this was a really terrible situation. But out of this came something really great, because this was a turning point for the country, and they decided then they needed to put more effort into disease surveillance. And so then they developed this pioneering integrated wheat rust early warning system, which is now the best ever wheat rust early warning system that we have anywhere globally. And so this wheat rust early warning system that came out of that terrible epidemic consists of a number of different components that they've developed in country. So they've developed these coordinated wheat rust surveys. They've got over 3 million subscribers to the phone app that's integrated in these surveys, so they can send out targeted alerts when they see disease in the field. They've also invested heavily in infrastructure and human capacity in rust pathology, so they can do the pathology tests of isolates. They can take isolates from the field, and directly in Ethiopia, they can work out which of their wheat varieties are susceptible to the isolates they have in the field. And then they've also developed these really fantastic facilities for screening varieties and fast-tracking the most resistant varieties for release, so they get into the farmer's hands as quickly as possible. And then more recently, they also developed and integrated these real-time disease forecasting models, working with the University of Cambridge in the UK and the Met Office. However, despite all of these monumental advancements that they've now made in Ethiopia, and the fact that this is really working very effectively at protecting the country from these kind of severe level epidemics, they were still missing a key component, which was the ability to define the individual strain of the pathogen that they had in real time. And so, of course, you can go out to the field, you can see that your field is yellow when you have yellow rust, you can see the pustules on the leaves, but they weren't able to determine which strain they had in the field. And it's very important to know which strain you have, as if you don't know which strain you have, you can't action the most appropriate control. Maybe the strain that comes in might be resistant to particular fungicides, so you need to know that information if you're going to action really appropriate control. And so that's where we then decided to move our field pathogenomics strategy over to this mobile Oxford MinION sequencing platform that is created by Oxford Nanopore, because then they would be able to run these MinION sequences directly in Ethiopia. And of course, this is not a new concept, so many people have used these MinION sequencers for in-field diagnostics and for tracking individual strains, and a lot of this work has been done around viruses and bacteria, and these are some examples from the human field, particularly working on Ebola and Zika virus. A lot of work has been done around this, and then more recently, of course, people have been doing this in plant pathogens, but again, focusing around viruses. And this is likely because viruses are easier to extract from samples and then genotype using these kind of platforms. Whereas we're working with a fungal organism, which comes with a whole bunch of challenges. So some of the challenges that we have of trying to do this on a small mobile platform is that this particular platform, this MinION sequencer, doesn't generate as much data as a big Illumina sequencing machine. And so because you get very limited data from this sequencing machine, it really does then limit the number of samples you can process. And also, fungi have very large genome sizes. So this method has been used extensively to genotype strains of Ebola, and this is shown in scale. Here is the size of the Ebola virus genome, and here is the size of our wheat yellow rust pathogens genome. And of course, this yellow rust pathogen, well, this is 10,000 times larger than an Ebola virus. And so you can imagine then, if you were to sequence the whole genome of the yellow rust pathogen, you could sequence 10,000 genomes of the Ebola virus in comparison. And so this makes it very difficult. But then, of course, you also have the added complexity that we're frequently working with obligate biotrace, as with this wheat yellow rust pathogen, and they can't be separated from their plant host. So if you were to just take an infected leaf sample from the field and then sequence it, you'd also get the host plant. And the wheat genome, well, that's a whole nother scale of size in comparison to the Ebola virus. And so it's very difficult to actually develop a method that you would get enough data to do strain-level diagnostics, because you could imagine that you would potentially take that infected leaf and you could sequence it on this MinION platform. And from the data you generate, you might be able to determine that, yes, there might be some yellow rust in there, but you already knew that because you saw it in the field. But to determine which strain you have, well, the strain-level information is going to be completely drowned out by all the other information in that sample. And so this was a really big challenge, and we needed a new strategy that could enrich for pathogen DNA to allow us to thereby sequence specifically the pathogen and not the plant host, but still go from those infected samples from the field, because that's really the quickest way to get your answer. But it also needed a method that could define individual strains and generate enough data per sample, but with only a small amount of data per sample, because otherwise you'd only be sequencing maybe one sample on a flow cell. Well, that's really ridiculously expensive. So you want to make this cost effective. You want to generate then the smallest amount of data per sample. And so the work I'm now going to describe was carried out very much in collaboration with Dave Hodson at CIMMIT and also Tedessa at EIAR in Ethiopia. And so to get around this, what we did was we looked at all the data that we'd generated from sequencing more than 1,000 samples using our field pathogenomics strategy, and we found that in general around 14,000 genes were turned on in the pathogen when we sequenced an infected leaf. But when we looked at the sequence of all those genes and we compared them between strains, we found, of course, that most of those genes were identical in sequence between strains, and that actually only a small portion of genes were different between strains. And we were actually able to differentiate just a core set of 242 genes. And if we just analyzed the sequence of those 242 genes, this was sufficient to define individual strains of our yellow rust pathogen. And so then we decided we could go forward with just doing a simple PCR of those 242 genes in a multiplex fashion. And then just by sequencing only those 242 genes, we were able to then define the individual strain that you have in your sample. And so we then had to convert, of course, our field pathogenomics strategy into this new platform, which we've called MARPL Diagnostics, for mobile and real-time plant disease diagnostics. And how this method works in practice is, again, we go out and we identify our infected leaf samples. We then do a quick and dirty DNA extraction, a PCR, and stick what comes out of the PCR machine on the mobile sequencing platform. We then have an automated analysis pipeline that allows us to then define the individual strain of the pathogen, and this whole process can be carried out in just 2 days of getting samples from the field. And so this now does provide real-time disease diagnostics at the strain level that can be integrated directly into that early warning system in Ethiopia. And also, of course, this provides a roadmap that can be applied now to other complex fungal plant pathogens. And also through this process, we developed a mobile lab system. So all the components, we put them all within a suitcase, and we developed a system so that you wouldn't then require access to any other additional sophisticated laboratory equipment. So everything that we developed had to be as simple as possible to make sure that the largest number of people could access this methodology. And so this created a truly mobile rapid response laboratory. And so how does this actually compare, of course, to existing methods? Well, unfortunately, you still have to carry out pathology, which is very, very lengthy and labor intensive. And so through the pathology analysis, what you do is you go out and you collect strains of the pathogen from the field, you bring them back, you then multiply them until you have enough spores to infect lots of different wheat varieties and work out which ones are susceptible to that pathogen strain. And of course, that whole process can take up to a year to get the real information that you need. However, what's really great about wheat yellow rust is that the genotype is very tightly linked to the pathology of that isolate. And so you can imagine then if you have an isolate where you've carried out your pathology tests and you know which wheat varieties that isolate can infect, and then you take a new isolate from the field and you genotype it and they're identical, it kind of gives you a proxy as to which wheat varieties are likely going to be infected by the new isolate you've taken from the field. And so this is really great because we can use genotypic characterization as a rapid method of getting a proxy as to which wheat varieties are under threat. And traditionally, there are a number, of course, of existing genotyping methods that are used currently for genotyping strains of the wheat yellow rust pathogen. And this includes, of course, checking the length of SSR marker regions or indeed the sequence at SNP markers. And unfortunately, most of these methods take a very long time, so you do have to grow up these isolates often to get enough DNA to run these methods. And also, these methods are only executed at specific laboratories around the globe. And for instance, in Ethiopia, there are no labs that can run these methods. And so you have to ship samples to centralized specialist laboratories to do your genotypic characterization. Whereas in contrast, this MARPL method that we developed, it is very, very flexible. It's also not dependent on predetermined marker sets, as you can add in genes of interest at any time. It's not like a SNP chip that's completely fixed. You can add and take out genes as much as you want. And you can carry it out pretty much anywhere. And because it's very quick and it doesn't require much hands-on time, and of course, personnel time is the most expensive element of all of our grants, it means that overall it equates to a much lower cost. And so we have actually tried this method in Ethiopia, and we ran a trial at the end of last year, and we developed a very simple DNA prep. And we carried all the DNA prep and the PCR out using no cold chain storage, because that's also an issue when you're working in developing countries. And the whole method was set up to be independent of the internet and independent of stable electricity. So the PCR machine, for instance, runs on a battery pack. And the analysis pipeline is fully automated, so anybody can pick it up. It's not something where you need high-level computational expertise. You can definitely pick this up. Anybody can do it. And we were able to show that even in the back of a car, we could go through this method in just 2 days and get our genotypic results about which strains we had in the field in Ethiopia. And so that data went immediately into the wheat rust advisory that goes out to stakeholders as part of the wheat rust early warning system. Okay, so then the final thing I just wanted to briefly mention was we've been discussing about how we actually make all our data accessible. And so I think it's really important that when we generate lots of data like this, that we provide it not just by dumping reads into a public database, which we should do, but also think about how we can make this data in an accessible format to everybody, regardless of their expertise and background. And so one of the things that we did was when we started generating this project, we thought about also how they are displaying this kind of unified data sets within human diseases. So we took our inspiration by utilizing the MinION platform for genotyping from what's going on with human diseases. So we also looked at what they were doing for visualizing data. And so one of the fantastic tools that we came across, also at a meeting that we were meeting with the creators of this tool, was NextStrain. And so NextStrain is used to look over time at mutations that occur in, for instance, the influenza virus, and then to track the distribution of those individual strains. And so we decided that we wanted to also utilize this fantastic tool, and so we set it up for the data that we're generating through the MARPL project for our yellow rust pathogen. And so this is an interactive tool which allows people to visualize the data that we've generated. We've got within this particular instance now, it's got over 300 samples, I think, in there. And you can see that you can visualize it as a phylogenetic tree. You can play with the data. You can look at which particular strains you have. You can look at them based on which countries they're present in. And over time, as this database continues to be built up, you can then track the movement of individual strains across the globe. And so this makes our data not just available, but also accessible to people. And of course, if anybody has any suggestions for improvement or other data visualization tools, then I'm always open to new ideas. Okay, so in summary, I think today I managed to convey that this field pathogenomics method that we started a few years ago now is a very powerful tool to assess the pathogen and host interactions directly from these field collected samples. And that we can generate data from that to identify genes in the host side of the interaction which are involved in disease susceptibility that can then be utilized by our breeders to then potentially improve disease management and provide them with a new source of resistance. And that this MARPL diagnostics technique, which is quite new, it was only, we actually only published the paper a month ago. So this is a new mobile genotyping platform which has been deployed in Ethiopia to enhance disease diagnostics capabilities. And so then I have to, of course, acknowledge all the people who actually do all the work. So my lab here, they're the guys who really pick up the pipettes and do all of the work in the lab. And then, of course, our collaborators. We can't do any of the work that we do without the fantastic support that we have from our collaborators. They're really quite amazing community. And, of course, our funders. And thank you very much for your time.
Thank you, Diane, for a very inspiring presentation. So we'll immediately go to the last presenter of today. So Kim Hammond-Cossack will be telling us more about databases, data sharing, and availability of data.
Right, well, first I'd like to start by thanking the organisers for inviting me to this very interesting meeting. And I'm now going to tell you a little bit about a database, a FAIR database that we're using in combination with bioinformatics to actually improve not only plant health, but also human, animal and ecosystem health. And this has been put together by my colleagues at Rothenstein Research, Martin Urban and Elaine Kuzik, who've been long-term members of the team. I have to also thank the BBSRC for very long-term funding that supported this, and more recently we've actually joined the Elixir Data for Life project as part of the UK node, providing data for agri-genomics experimentation. So the database is called Phybase. It stands for Pathogen Host Interactions, and this is very much a multi-species database which is used to understand the mechanisms that are underlying pathogenicity. So how do pathogens cause disease? And then from that, take that novel knowledge collectively and then come up with exploring new ways to protect crops, humans, animals and ecosystems, their health. So what is Phybase? Well, it's a web-accessible relational phenotypic database that catalogues genes which the experimental community have actually tested for their role in pathogenicity. It comes about by manual curation by both domain and species experts, and in each case the entry in the database is supported by a peer-reviewed literature. So the type of literature that we're surveying each month involves pathogen gene deletions or modifications or silencings and comparing strains with these modifications to the wild type. As I said, the scope of the database is quite large, in different pathogens and host species. And what we do as a result of interacting with the community has been we not only include positive data where a manipulation of the genome has led to a reduction in pathogenicity or a loss of pathogenicity, but we also include experiments where there's no change in the phenotype. So that's what we call bi-negative data. It's freely accessible at that particular URL, and it's been available since 2005. And we now know that the database is regularly used in over 130 countries, over 6 continents, and we have about 12,000 users a year. If you don't like the database format, you have the ability to download the data freely at any time as well and use it in whatever format you want, which helps companies who are interested in this topic. Okay, so our motivations really were that plant pathogen host interactions are changing all the time. They're often every year. Major crop disease outbreaks are reported, or they can be human or animal epidemics. They can also often arise as a result of emerging fungicide problems or anti-infective resistance. And just looking at this gallery here, back in the 1800s, we had Irish potato famine, but more recently we are worried about wheat blast, coffee rust, Asian soybean rust. There's always another pathogen on the horizon that's going to end up making the headlines, and we just don't know where it's going to happen and when it's going to happen. So as I said, Phybase is multi-species. We particularly wanted to do that to try and understand generic natures of disease-causing ability. Our focus is fungi, protists and bacteria. As others have reported, viruses are already well covered in other databases. At the moment, we've fixed our number of species that we curate at 250. And we have a focus on agricultural crops, horticultural crops, commercial tree species, whether it's timber or fruit, the model species, and then emerging pathogens. We also focus on the animal side, on key human pathogens, where the organism under test is used on the non-pathogenic, on the non-mammalian species, such as insects, and then we don't extend very much into plant-attacking nematodes or insects. Our schema would allow for this. And just to give you an idea here of the top 15 in the latest database release, which happened just a couple of weeks ago, we have serial infecting pathogens like Fusarium graminearum and Magnaportha oryzae and Xanthomyces oryzae. I see that number 3 we've already heard about this morning, Ralstonia solanarum. In the second row, we have human pathogens like Candida albicans, the fungus, opportunistic bacteria like Pseudomonas aeruginosa, which will both attack humans as well as plants, very wide, broad host range like Botrytis cinerea. And then on the bottom, an increasing number of medically important pathogens. So just to give you an idea of the data that's in there, it's come at the moment from a little under 3,500 references. It contains information, phenotypic information and biology on over 8,000 genes that have been tested in more than 13,000 interactions. As I said, the pathogens are capped at about 250. But with name changes, they sometimes end up a bit more in subspeciation. And we're also cataloguing the chemistries which are involved in fungicide control or anti-infectives. Pathogens themselves, the database is 75% eukaryotic, either fungi or protists, and 25% bacteria. And the hosts are about 60% plants, 50-50 cereal and non-cereal, and the other 40% are animal or human pathogens. So it's quite, as I said, a wide-ranging database. And again, you've seen slides like this. What was our motivation, really? Well, that is because if you are actually keyword searching the literature for looking for genes associated with virulence, the number of publications is going up and up over the decades. So the first clear publications were back in the 1980s, where the first bacterial A virulence genes were cloned, and the first fungal A virulence gene expressed in plant in the 1990s. But then, since genome sequencing really took off in the early to mid-2000s in the public domain, and then all different types of sequencing techniques came out, then there's an increasing number of functional genomic studies that are taking place and are reported. So what we do is this. We basically have every month, we go through and do a keyword search to identify new papers, which are generating the types of information we want to catalogue, and we're even now trialling machine learning to find those papers. We then review that list, and that's done actually in India now, a company called Molecular Connections, based in Bangalore. We then review that list, and then we add submissions from actually individuals around the globe who actually note these types of papers. And then those papers are curated at 10 per week, and we're taking out enormous amounts of data out of there, 81 different data points for each gene. That's coming from the main text. It's also coming from the figures and the tables, so it can't just be gleaned by text mining. And then we do 2 data releases a year. So we come through the actual curation process, and then it goes into a MySQL relational database, and then it eventually comes through to the web pages. And this is what it would look like if you want to go and look. On those ones, we have a number of ways of exploring it. You can start off very much with a search. You can put in a gene of interest, a species of interest, a type of protein of interest, and hit search. Or you can do a more advanced search where you're doing combinations of genes with species and the type of phenotype you're looking for. In point 2 there, it will bring back information on the gene of interest, what the protein ID is, what type of pathway or function the gene may actually have, give you information about the pathogen species straight away, the strain by which it was developed, also the host which was tested on, information on the publication, here by 4, and also on the right-hand side here, the type of experiment that was actually done. So in this case, it was an experiment where the mutation looking at a single gene led to reduced virulence. You can also look and see in 4 there if there's actually a related gene involved in the process, or it might be a double mutation, and see what the phenotypes are there. So that's one way of analysing, looking for data of interest. The other is to use the BLAST-B function up there, where you take your sequence of interest, which may come from a genome experiment, or it may come from a forward or a reverse genetics experiment, and ask, is there an entry in the database that's is sequence related? And in this case, the BLAST result is a bit different. You get the usual top bit where you show the alignment at the protein level. But what happens actually in the header when it comes back, you can actually see that the first entry there is a magnaportha gene, and the phenotype, when it's knocked out, is reduced virulence. So you have a phi number on there, which you can then go and look in more detail by clicking on, and you know straight away that this gene is required for virulence. And in that top list there, you can see that 3 related sequences in 3 different fungi are all required for virulence. Okay, so what does it use for, and why might it be of interest to this community? Well, when we set up the database, we had particular interests in it, but we found that over time, people have found this database, and they're now using it for all different sorts of purposes. And because we don't provide many tools with it, what we do is we provide the publications that have cited Phybase use, and then it gives people other ideas what they could do with the data in this database. So as I said to begin with, there's a lookup function to look for gene mutation phenotype relationships, along with the BLAST tool, and you can fully download all of the data, not just from a search. You can download the whole data set. I'm going to go in a minute to explain how we actually provide phenotypic annotation for genome browsers, such as Ensemble Genomes and FungalDB. But you might also be in a situation where you have a newly sequenced pathogen genome, or indeed a series of genomes, and you're looking at the variome, and you want to put annotation onto that data set. Alternatively, you may have an RNA-seq or a microarray data set, and again, you want to look in there and track the expression of particular virulence genes known to be associated with the pathogenic process. Or your gene may come from a forward genetics experiment or even a GWAS analysis, and you want to see what's the potential of linking this sequence to pathogenicity. Or you can use it for comparative genomics and phenomics, such as comparing conserved themes as against species-specific, or in silico predictions of what might be candidate virulence genes by doing protein-protein interaction networks. So it's available for all those types of sources. But let's take the simplest one there. Let's try and provide now phenotypic annotation directly on genome browsers, which can be easily found. Okay, so to do this, we wanted to come up with a species-neutral terminology so that all genes could be explored across all multiple species. And so we identified and published 9 high-level terms which would describe any interaction outcome. The first 5 we would term are the virulence terms, which will be loss of pathogenicity, reduced virulence, increased virulence, unaffected pathogenicity, or effectors, which are these transferred entities in an interaction. We also have things on antagonisms, essential for life, and then resistance or sensitive to chemistry. So these are not covered by Go terms or anything like that, gene ontology terms, but because they're high level and species neutral, then they can be used to compare mechanisms across species. But to give you an idea here, how we use these high level phenotypes, we could define an interaction as the function of one gene on one host on one tissue type from one publication. And that's what would be given a Phybase number. So in this case, we have a Fusarium graminearum and a mutant here called Tri5. If it's tested in an interaction on wheat ears, the interaction outcome there is reduced virulence. If it's tested in a wheat seedling assay at a much younger state, again, it is reduced virulence of the mutant. But in the third assay on a maize cob, it's reported in the literature that's pathogenicity unaffected. So you can therefore narrow down by comparing interactions which things are narrow in their function and which things are actually far wider. So this gives you a summary of the data that's actually in Phybase. And you can see that the main category there is indeed reduced virulence. So when a gene is modified in some way, you end up with less disease. We have nearly just over a quarter of the data is unaffected pathogenicity, which is a good comparator. But we also have other categories which are in growing interest now in the community, which are effectors, these transferred entities at 16%. And also an increasingly growing category of called increased virulence. So when you take away the function of a gene, you actually lead to more disease. And so there's some negative regulation in the system. As I said, the community asked for this to happen, and it actually is now very good for comparing what genes and pathways are required in one species, but not required in another one. So how do we integrate this Phybase data with a genome resource? So what we do here is we have Phybase providing the phenotypes and the experimental information, and then this is actually pushed every 3 months to Ensemble genomes, where they actually can then display that information on the reference genomes for each pathogen, also on the variomes, and we can now take advantage of their very sophisticated comparative genomics tools, such as like the CompareA tool, to explore closely related subspecies. So to actually do this mapping, we either directly use the UniProt IDs to do the mapping, or we use sequence similarity, and that has to be 100% identity with BLAST-P within subtaxa. And then you can see below that we are now already projecting our curated information on over 1,100 genomes across fungi, protists, and bacteria. And therefore, it allows also the use of a second tool called the Biomart tool to search for annotation for genes of interest. To give you an idea, now you're on a genome browser. In this case, it's Magnaportha, the rice blast fungus. And as you go along, you can see the models predicted there, gene models in red. And if you see a color-coded gene in green or in orange, you know then there's some annotation from Phybase. And you just need to click onto that gene, and down will come a little drop-down box, and it will tell you straight away that the green gene there leads to a loss of pathogenicity when it's taken out, and the orange gene leads to a reduced virulence. So that those 9 high-level terms are color-coded on the genome. You can also use this Biomart tool to find it. You go in the header of Ensemble, and then you work along to the third tab called Biomart. Within the filters, which I've circled there, you just click that, and then you come up with this page. And you can see the third point down there, you can actually then go for pathogen phenotypes, and you can select your host, your type of interaction outcome you're looking for, and also your experimental conditions. Then run a search, and it brings back results for a particular pathogen or group of pathogens. And when you look in detail there, it will tell you straight away for these particular genes, I was selecting there for reduced virulence, gives you the Phybase number, how the experiment was done. And then you can take that information and download it in various formats. In this case, I chose Excel, and then work on it in your own local PC. Okay, and then you also can use the CompareA tool that they have, and then you have your gene of interest now, and immediately it's put inside a phylogeny, and you can look up for the related sequence in closely related species, in this case another Zymoseptoria and a Ramulera and other serial infecting pathogens. So it gives you an enormous amount of information in a very short amount of time on a gene that's been linked already to pathogenicity. Okay, and then what we've done more recently in the last couple of years is actually to further enhance the tool by saying that we want now to actually involve the authors of the publications actually in helping the curation process, because quite frankly, they know the most about their data. They generated it. And so we've come up with a tool called PhyCanto, which allows authors now to curate their papers. Those entries are then checked by species experts, and then that will go into Phybase. So that will help with the data deluge that's coming from the peer-reviewed literature. Then on the right-hand side, we now have that data sets going through into Ensemble fungi, bacteria, or protists at Ensemble. And what we've also been doing is curating the first host targets of pathogen effectors, and this will very soon be arriving in Ensemble plants, so you can actually find out exactly what are the targets of these effectors which are influencing the interaction outcome. And this is to give you an idea of what we're doing, this PhyCanto tool that we've been developing with the University of Cambridge and the POM-based community, the fission yeast community. What you'll need to do is basically take your PubMed ID, put that in, and then within about 15 or 20 minutes following a series of pages, you can actually then completely curate the genes you were involved in, the type of experiment, the phenotypic outcome, and then submit that for review by other experts. So that should help to actually ensure that all the data is successfully curated. And I have to say that I have to thank the Phybase for a new grant that's just started to actually further develop this tool and the ontologies that need to be developed to fully be able to populate the various fields. Okay, so I've told you a bit about the database, maybe a few a bit too much data, but I now want to just consider in the remains of my talk, how could Phybase be used, uses that are relevant to your organisations, in the era of metagenomics. So I've picked out a few areas that I think may be of value. And the first is in emerging infectious diseases. And I can imagine a typical workflow is that you identify a problematic pathogen species taxa, possibly by ITS sequencing or another species-specific primers. You then decide that you'll undergo full genome sequencing, annotation, and assembly and predict the open breeding frames. And then you come to the dilemma, do you do a limited gene set, or do you go ahead and do full genome annotation? And I might suggest that it may be best to actually, in the first instance, go for a limited gene set evaluation. It will be less time and probably more cost effective. And one of the areas to look at is this hypervirulence category, which is in the database, which currently is at 4.6%, where you knock out a gene or change it slightly and it leads to more disease. Now, we're noticing that over the last 5 or 6 years, this category is gradually creeping up in the database, and already there's over 500 genes reported from over 100 pathogenic species when modified leads to more disease. And amongst the plant pathogens, there's nearly 200 genes across 49 pathogenic species that are leading to increased disease. And it's also noticeable that certain host species are also having more entries of this type, like rice and the grapevine. I thought I'd give you an example, a couple of examples here, what we've got. So the first is indeed Magnaportha, the rice blast pathogen, and so far there are 10 hypervirulence entries there. Rice blast causes both a leaf and a neck disease, lowering yield. And one of the key aspects of invasion is after spore germination, the aprosorium is formed, and then that punches a hole through the cuticle of the rice epidermis. And it's notable that if you actually remove an intracellular signalling gene called RGS1, which is that actually you end up with more Aprosoria formation, those dark blobs, compared with the wild type. And this leads to more disease. Also, these mutants produce more asexual spores, so there's more potential for dissemination between plants. So this is clearly somehow involved in negative regulation of a pathway controlling Aprosoria formation. Now, you can note from your own knowledge and also from reading the literature that many pathogenic fungi need to produce Aprosorium to infect plant surfaces, for example, the powdery mildews, the rusts, and the Colloquium species. So immediately one can exploit that knowledge on a generic lifestyle theme to ask the question, if homologs of that in an emerging plant species, have they got this gene? Is it in the wild type form? Is it in the mutated form? Could this be the cause of the emerging disease problem? The second example is a bacterial example. We've already heard Zaire fastidiosa infecting grapevines, but the example here I have from olives, and there's already 13 examples of hypervirulence that have been reported in the literature. On olives, it came in 2013 into Europe. And the example here, it's a vascular attacking bacteria, is where they were exploring outer membrane vesicles produced by Viella, and by blocking they produced them to block the interaction with various surfaces, including the xylem vessel walls. And these OMVs stop bacterial adhesion. So in the life cycle, basically, when the bacteria is only producing very low numbers of these outer membrane vesicles, it remains static and can actually be acquired by its insect vector host. But as soon as the pathogen switches to high OMV production, then the bacteria becomes systemic and spread through the host. So basically, there's a mutant here called RPF1, has been reported, which is disrupted in quorum sensing, and therefore it has an increased OMV release of 5-fold. As a result of that, you end up with less adherence to plant surfaces, the bacteria becomes more systemic within the olive plants, and then you end up with more disease. So again, this is another exploitable generic lifetime theme that would then indicate particular genes that could be tracked for variation in populations and in particular samples and where there's been disease outbreaks. Oops. Okay, now another way that new diseases can emerge is by the transfer of interspecific virulence gene transfer, and I think one of the classic examples here is in the serial infecting fungi, where it's now noted that in about the 1940s, the transfer of a small chunk of DNA, around about 11 kb, contained a particular gene called TOXA, and that meant that the gene was able to transfer from Septoria nodorum, which causes a blotch disease on wheat leaves, into a proto-species, which then led to basically the arrival of tan spot called Bipyronophora. And the target of this gene, TOXA, is indeed TSN1, which is a susceptibility locus. So once you have that sort of information, you could actually say, well, if you now remove TSN1 from the wheat population, you will actually then enhance resistance to both of these pathogenic species. Another way in which you can have newly emerging disease is by the acquisition or modification of secondary metabolite clusters. Also in our database are a large number of pathogen effectors on their first host targets, and for those who are not too sure, effectors are an entity produced by a pathogenic species to facilitate the infection process, either by activating or suppressing host defences or other types of host responses. They can be produced into the apoplast, i.e. extracellular from the pathogen, or they can go into the cytoplasm where they may directly or indirectly interact with plant proteins. So now, as a result of interactions with the community, we now actually have 16% of the database, over 2,200 entries on effectors, coming from 83 species. And as I said, we're starting to curate the first host targets that will soon be displayed in ensemble plants. So most effectors are species or pathogen strain specific, and therefore they would be maximum discovery of effectors would come by exploiting the variomes, which are displayed on genome browsers. But there's also instances where some of these effectors are actually essential for pathogenicity, and it is very good to actually know which ones are these essential for the disease-forming process. For example, some of the chitin-binding ones like ECP6 and 3-lyse-M. So what can you do with effectors knowledge? Well, one of the contributions that Phybase can make is via plant breeding and the use of effectoromics, which effectoromics is a high-throughput functional genomics approach that probes basically plant germplasm for lines that can actually detect effectors and therefore may possess particular resistance proteins. So in effectoromics, you would invariably have a panel of plant species and a panel of effectors, individual ones, and then you test them in this matrix. And so you'd be looking for identification of specific R gene functions. You'd be distinguishing functional redundancy. So in the middle here, we've got three effectors which are recognized by two different host species. You can pinpoint recognition specificities, and therefore you can assist in R gene deployment. And it's been particularly applicable up to now against Phytophthora infestans, the late blight fungus, oscillating between the true host potato and also a model species, Nicotiana bienfemiana, and then using the techniques of agroinfiltration and agroinfections. And what's really happening here is the breeders aim to maintain the widest possible repertoire of disease resistance genes in elite germplasm. So effectoromics, once they've identified, could be used in a lot of species to actually then identify germplasm that may give you resistance to these emerging threats. Another area you may not have considered is actually discovering of novel antifungal, anti-infective targets. And what's also rising in the database is the number of times that lethals are reported in experimentation. And for plant pathogens, that's over 100 now. And this is an example where when you try and knock a gene out or modify it, you actually don't recover any transformants at all, whereas in other genes you've selected, you do find a phenotype. And then this new information about essential for life genes, they can be nominated for novel intervention targets that can be used by biotech companies or chem companies or bioscientists to find out why that gene is essential for life. But you can also take the proteins as well from there and put them into chemical screens to try and identify natural products or synthetic chemistries that may interfere with their functions. And it's highly likely that essential for life genes are indeed conserved between species. So what we can gain already from communities like the Candida albicans community, what they are learning about its lethals, can some of that be projected onto other Ascomycete fungi? And then if we turn to pangenome analysis, we've already heard about core genomes versus variable genomes. There's a lot of potential now to be looking at what is actually happening between both the core genome and the variable genome when you're comparing historic isolates and then recently emerging problematical isolates. Secondly, you can also look at what's happening when we've got genomes from older strains when there were previous control practices and now you're comparing it with new or existing control practices. For example, a switch in a region maybe between fungicide use and biocontrol options plus resistance gene deployment. So what parts of the genome vary, and there from there, what would you actually decide to explore and monitor for changes? So this is the whole idea here about pinpointing the genome evolutionary changes. But I do want to point out here, it's not just about the gene sets that are happening here, which are in the core genome and the variable genome. It's also what's the genome location in the genome of these varying sequences. And just to give you two examples, you may have a genome where you've got minimum repetitive DNA or transposons, but through experimentation, you know that across the genome, you've got areas which are full of SNPs and others that are not. In those areas where there's a high number of SNPs, you've got a high recombination frequency. And then when you've actually done some comparative analysis, as in this example here, where the regions which have high SNPs and recombination, it's also the regions where the genes are only found in that species and not found in other related species. This happens to be chromosome 1 of Fusarium graminearum I work on, but you can really see that if you were interested in coming up potentially with taxon-specific sequences, you'd be going into those higher recombination areas with SNP densities. The other scenario is where you have a genome which is rich in repetitive DNA or transposons, and they're often available in blocks in the genome. And what's happening here is that you often find that species-specific genes are neighbouring those blocks of repetitive DNA, or they are indeed where resides in the genome the small effector genes. But what you find is if you use repeat masker software, this will actually obscure the importance of those gene sequences. So it's important to go back and redo analysis without repeat masker to really recover the full repertoire of these small effectors or species-specific genes. And then you can also take information from well-explored species, the genomes, and extrapolate that onto underexplored species. And by doing that, you can take advantage of taxonomy or the lifestyle relatedness of the species. So here I've just come within the Fusarium. You have the serial infecting ones and the non-serial infecting. And I've given you the range of interaction types there that you see. So for Fusarium graminearum, over 1,500 interactions are reported, whereas for other species, 5 species, there's only 21 interactions. Likewise, for Fusarium oxysporum, it's only a sixth of what you've got for Fusarium graminearum, but there's still significantly more than Fusarium sonale. So you may well think you should just be comparing within the serials or the non-serial species, but then if you look at the infection biology, you can turn around and say, well, actually, Fusarium graminearum does have a vascular associated phase of its infection, therefore knowledge from Fusarium oxysporum could be valuable to further understand infection by Fusarium graminearum, or vice versa. You could say the small phase of vascular infection could actually then inform on the genes required for disease-causing ability by a vascular attacking pathogen. Okay, and then my final example is where we are at the moment in our lab, and that is on predictive biology and network analysis. And this, I think, is a great potential to bring together all the data sets that are available for pathogens and all that experimental knowledge. And a lot of this is going to involve graphical visualization analysis to explore protein-protein interaction networks that have been that can get predicted, and then from there potentially reveal the Achilles heel for particular problematic species. So we've been generating PPI networks, as they're called, based on interlogs and domains. These typically contain between 3,500 and 8,000 nodes, that's protein nodes, and then the edges which connect them somewhere between 150,000 to 450,000. And then they can be visualized in software like Cytoscape. And we've done that for about 15 plant pathogens now. I have to say that it is quite slow, and I take to create 15 of these pathonets, as we call them. FiNets has taken a year. And what you get down to at the end is little interesting sub-networks where things you think are related to pathogenicity. So in this case, I've given you a diagram of 15 proteins with their locus IDs. One of them is predicted to be a target site for a small interfering RNA. Another one is known by gene knockouts to be required for virulence, but the rest of them, there's no biology on them at all. They don't have any, or they have minimal Go annotation, what their functions might be. And so, as I said, it took a year to come together to create these type of networks. But there are new tools out there now, and one of the one I'm particularly interested in is NetMiner, which is being generated at Rothamsted, and that's a graphical visualization analysis. And if you take that tool, last week when I was doing this, within 45 minutes, I'd actually already got considerably more information on 3 of those genes. So in this case, by taking a gene of interest, within NetMiner, it was found that that gene actually had relationships to 3 others. Those red blobs are related gene sequences. And already in the literature, 3 of those genes had already been knocked out and led to a reduced virulence phenotype. So possibly, by knocking out that gene, we might see an effect on virulence. The second example, where in NetMiner, again, there was a gene, a related sequence had already been knocked out and again led to virulence, reduced reduction in virulence, and likewise a third one over here. So once you've actually got information collated in networks, you can very readily add in extra information and increase the value of the knowledge you've already gained from a much longer-winded technique. Okay, and my final slide is thinking about the universe in which you're working in. This is my new universe, the Phi universe, where we're very much following the FAIR data principles of findability, accessibility, interoperability, and reusability. So we're not fragmenting our data and our knowledge in any way. As I said, below the line, these are things we've already got going. So Phybase data, phenotypic data goes on into Ensemble. We also now have it in FungiDB. We are taking information at the moment from UniProt, but there's no reason why over time we couldn't actually provide information to UniProt if we could actually come up with a way of being interoperable. Likewise, we're pretty soon going to be presenting our data sets into the JGI microcosm database. But above, we've got the possibility here that actually individual research, there's no reason why they now can't actually add information almost directly into Phybase. And then there could be the possibility that individual species databases or collected species database information could flow in. And then there's important knowledge which at the moment is fragmented, is not available. So CABI has a wonderful Thesaurus and species invasive species compendium, but that information is just within CABI that could actually come in and actually give people a lot more information about lifestyle host ranges. So I think if you look at the universe and think how can you make it fair, then you can see where you have spent time curating information to a high level, it can immediately blossom out and be a much bigger data set if you can only connect to a much wider range of databases and resources. And very finally, I'd like to very much thank all the members of the team involved at Rothamsted. We have got a large list there of bioinformaticians, wet biologists talking together. We have the POM-based team at Cambridge who've gone from a one-species curation tool to this multi-species curation tool successfully over a 2-year period. I have to thank the individual species experts globally that check our entries to make sure that it is gold standard. And then there's the Phybase team actually at the EBI who's involved in putting it into Ensemble. And our collaborators in Elixir and our curators of molecular connections in India. So thank you for your time, and I'm happy to. Yeah.
Maybe you can stay here
Thank you.
for
Yeah,
the discussion.
Yeah, thank you.
So, Kim, thank you very much for your lovely presentation. I would like to invite Diana back on the stage and also Baldicera for us to continue to the final discussion session and maybe to so obviously we can continue where we left off in the previous session. Also, if some of the presenters of this morning session are willing to come on the stage, of course you're free to collaborate from over here, or ask or pose intriguing statements from the audience. That is fine as well, of course. So in the afternoon session, we've been hearing about on-site applications of new sequencing technologies, and also how connecting databases and fair data sharing principles can aid in further unlocking all the potential of these powerful tools that we've been building and curating. So first I would like to open the floor to you, the audience, if there are any questions to the presenters or issues to be raised. Oh, Baldi.
Thank you, Bart. More than a question is an invitation. So first of all, I would like to thank Diane for the beautiful presentation. It's the second time I see your presentation, and I couldn't imagine that I would be amazed the second time. You presented us the We Trust early warning system. I guess it's a dream system for our community. And I would like to inform you that there is a incoming Euphresco project on system for awareness, early detection, and notification of organisms harmful to plants. And the idea of this project is to map notification systems around the world, or at least in the participating countries, and to see how to drag information from these systems to inform the national plant protection organizations, and how to use these systems to raise awareness. And so the coordinator of the project is Ilvo, and I think your experience would be much appreciated. And maybe, Chris, you want to add something on that?
I think I'm more than happy to help, so anything that I can provide, I think guidance would be fine.
So maybe a question to you, Diane. So regarding the early detection system that you've built together, so in On-site testing procedures. So how have you taken care of process controls and validation to make sure that what is happening in the field actually reflects what you're looking for? Yeah.
So I think, so for the field pathogenomics work, we worked mainly with breeders and agronomists and also ourselves and with collaborators. So they were all quite clued up on how to take the samples, and we had obviously best practice, and we had instructions that everybody followed. Sorry.
Use this one.
Yeah. So we had instructions that followed, that were very straightforward for people to follow. For the MARPL work, working in Ethiopia is challenging, and we've set up now, well, we're in the process of setting up 5 key hubs where we will provide all the training required, but I mean, it's not a complex process, so it should be quite straightforward for people to pick it up. And we always provide good documentation for everyone. And one of the great things with the MARPL work was, you know, even the equipment list, you can just download this from our website. It's, you know, everything you need should be there, and if it's not, then tell me, and it will be. So, yeah.
So I think, especially with the visualization tools that you've shown, those are really powerful in bringing the technique in the hands of also labs that have less experience with hardcore bioinformatics and other stuff, but do have experience with wet lab molecular techniques. Are there any questions from the audience? So what we were hoping, oh yeah, Robert.
Question for Diana.
Yeah.
Do you think that's the way to go for Is this working? Yeah, it's working. Do you think this is a good way to go to put a part of the main bioinformatics that you need in a kind of a user-friendly form so that everybody can use it?
Yeah, I mean, I don't know if it's, yeah, we're working. So I'm not a bioinformatician by training, but I'm quite capable of running all this analysis. I don't think that the level of bioinformatics that we do is particularly difficult. It just requires that we invest in training people. So I spend at least a month of my year, every year, training people in using bioinformatics in various different locations around the globe. And so I don't think that they're inaccessible. And I think people always get a bit scared because maybe they haven't looked at a black screen on a computer before, but it doesn't mean you can't do it. And I do think that it's good to make things as easy as possible, but I also think it's important that people understand, even if they use the simplistic version, that they understand what's going on behind it. So at least they've had some experience and some time one-to-one with people in training courses to understand a bit more about what's going on. But it is nice to be able to have automated pipelines where you press a button, but it is good also if you can fix them if they did break. So you need to know a little bit about how the pipeline works.
I'm just interested if is this approach already used in Europe for forecasting systems or for checking if there is a resistance development towards fungicides? Because I'm not sure. I think there is another university in UK working also on that for forecasting purposes. I was not sure if you are linked or not. Is this already used in Europe or?
So we have most countries in Europe have their own surveillance systems, and we've actually got a project specifically around wheat rust, which is run by Aarhus University in Denmark, and that project brings together all of those organizations across Europe so that we can unify some of the systems that we're using and share germplasm for screening and things like this. So actually in a couple of weeks I will run a training course with people within that so that they can also learn how to use the MARPL system. So then that can be used across Europe.
It's not already applied. It's
not currently applied in Europe, but so the disease forecasting work is done by Cambridge University, and we work closely with those people also in the UK, by Chris Gilligan's group.
So your 242 genes, so or markers, are targeting the rust. So do you think because you work with an obligate, could be interesting to have some marker for the host at some point to have some genotype or species identifications?
Yeah. So I think that it's true that we'd really like to do that. So originally the project was funded through $100,000 just to get it up and going through a competition. So it was very difficult to do more than what we did at the time. But as we move forward, we've seen some really interesting associations in the natural environment through our field pathogenomics work between particular strains of the fungus associating with particular varieties and at different times of the year and things like this, which have been very informative. So we would want to make sure that we don't lose that information going forward and that we're doing also incorporate markers for the different wheat varieties. And we have actually got like markers available for the different wheat varieties, so they've all been run through these like large SNP chips, so it wouldn't be difficult to develop some genes that we could amplify to define different varieties of wheat too.
Yes, I was impressed by all the presentations, but I started this morning also with the issue that this is also to see how research and regulators can link up with their work, and we have heard some of the elements, but it may be good to look a bit more into this element of, and what is important for regulators is of course the diagnostics, and we have heard some elements also, but also on pest risk analysis is important for our regulators. And we have heard the database, we've heard all molecular tools. Perhaps there are some people who could, from more the MPPO side, how they see, or perhaps from the EPPO team, how do you see that this, what was presented today, links up to diagnostics by MPPOs or pest risk analysis?
Of course, it links with at least for sure for first diagnostics. You know, if you're not aware, this is a topic that we are trying to. We've been asked to develop guidelines on high-throughput sequencing used in plant health. So this is something we're going to tackle. I see a number of challenges, in particular in the framework of implementation in the lab, so about quality assurance, about accreditation, but also about baseline. What I mean with baseline, for example, if you find something, for example, if you use high-throughput sequencing for analyzing a sample at import, and you try to find all what's there, and it tells you there's a virus there. Okay, and so what? Am I sure that this virus is not present? So I think the baseline, what I call the baseline, and what Belgium is doing is trying to have an idea about what you have in Solanum, for example, is a good example. And we need that baseline. Because if we don't have that baseline, I'm talking, you know, for those of you who know Asterix, I'm a little bit like Obelix. I found in the potion, magic potion for quarantine when I was a baby. So I'm very biased on quarantine. But when I think quarantine, I always think, am I able to justify what I'm doing against the others, against the exporting country? So I don't know if this answers your question. It's very useful. I mean, we have to go towards there. We have to have databases that can be curated and reliable for the labs to do their job. But we also have to have this. I think it's important to have this baseline.
Yeah, so I have a follow-up question on that maybe. So would you then say that this baseline would be most important in virology maybe? Because perhaps in virology is an area in which there might be hiding much, much more than we've seen so far, which maybe is not the case for some of the other organism groups we're working with.
I was checking in, we were in a COS meeting a few years ago, and we were asked again kind of the same question. So what are the challenges from your perspective? And one of them is, yes, in virology, the fact that they may be there without doing anything, or they may even be there with doing what you're looking for, like in ornamentals, maybe. What you see there, the spots on the leaves, etc. So I think probably in virology, but I don't know enough about it. You know, in the EPPO Secretariat, what we say most of the time, it's because you teached us. So it's not us learning, it's you teaching us most of the time.
Yeah, I think for the, Marleen, would you comment on that?
So I really agree with Francois, and I'm really happy with the effort of ILFO and others with establishing this baseline for solanaceous crops. But who is going to decide what other crops or weeds or whatever trees is such a baseline will be constructed? And are we going to do that only in Europe, share the information with the baselines in the United States and Australia? Because actually that's what you want, to find out that the virus you're working on is the same as you're working on another pathogen, and then share this information. But how are we going to do this? That's my question.
I think partly this baseline is also meant, if an example Françoise gave, you find something at import, and of course we have to answer the question, is this a risk? And that's already difficult, but also, and that's often forgotten, is that it may be said that is commonly present in your country.
Yes, that's what I mean. But
And how do you, but that's per country, of course, different.
Yes,
Yeah.
but maybe per area.
Maybe per area,
But
yeah.
you only find out once you start doing nationwide surveys, but who's going to provide for that and who's going to pay for that? And in what, so Amy already gave really nice examples how you can approach that. But I don't see it happening for many crops.
Yeah. And it ties into what Guillaume also mentioned. So the challenges, the numbers are so incredibly high. The different hosts, all the different viruses, bacteria, fungi. And for instance, when you look at all the effort that has been put in the early warning system for reed rusts, which, if I don't know if I put it too bluntly, is only 3 species, maybe? Yeah, and obviously we have hundreds of examples, and many of those have not received any attention from a genomic perspective. And so there's lots of genomes still missing, how to establish baselines. So does this maybe require coordination at the IPPC level? That could be a question maybe, or Euphresco to mobilize the globe in harmonizing and coordinating these type of activities. So is that something that you could envision? How, as a community, a plant health community, tackle with these new opportunities, but also the new questions that arise from it?
Yeah, I mean, I think it's very difficult to always decide on the priorities. Obviously, the wheat rust was a response to the outbreak of, UG99 and the potential movement of UG99 and the consequences that could have had if it had moved very rapidly into other areas around the world. And so that's why they started setting up this wheat rust early warning system, which was funded at the beginning of the 2000s and has been continuously funded since then by the Gates Foundation and then also by DFID in the UK. So I think it is always difficult, and at the moment, of course, in Europe we have Xylella, which is becoming quite a focus, and we're all very concerned about that right now. But I don't know how we make an ultimate list, because you have to think about the pathogens that are causing problems today and also the ones that might be coming tomorrow. So how do you prioritize those things? I think it's always a challenge for us to think about that.
And knowing that most species won't cause any problems at all, unless they
Yeah, and then you wouldn't anticipate that coming. So, so I think it is really challenging. But there are clearly some which stand out as being potentially damaging. So, like wheat blast was an obvious one when it moved out of South America. You can see the problem it's caused in South America and then moving into Bangladesh. I mean, this is horrific. So it was very clear that would cause a big problem there. So energy is needed in that direction. And the same with Salmonella in Europe. And we need to put effort into that because of the implications. So, but I don't know how you make this ultimate list and how to put the money in the right direction.
Sharing all the data we have with each other.
Yeah.
Find out, it's Effector already, forget about it, no problems.
Yeah.
So maybe it would start indeed with sharing data, creating openness and overviews of what is encountered in national surveys.
Yeah. But it can be also difficult for some countries to be able to share because of the implications of if they have a new disease in the country. So this is also a challenge that we will have to face, and reality that we would love everyone to be able to share, but some people feel that they can't for whatever reasons regarding trade or so on.
So we have been talking about this problem that is more a system problem, where to put the efforts, because the technology is very powerful and we can apply to everything, plants, water, soil. So the question is, when you have a good tool, what comes first? But I would like to come back from the Euphresco perspective to the research questions. So the technology may appear perfect, but there are still questions open, and I would like to ask the question to all the speakers. So what are for you the gaps that are still have to be addressed in terms of technological gaps? And then we have been talking about the infrastructures, and what are also the gaps related to the infrastructures that have been presented here, or that can support the activities that were presented.
Maybe Kim, would you?
Yeah. I think from recent discussions we've had in the UK, one of the areas where even the big genome providers know they have problems is actually pan-genome analysis. They are doing just about okay with reference genomes and a few other strains, but where they're going to really run into problems is the tools that are going to be able to actually effectively analyse pan-genomes. How do you actually make it in a readable format or an analysable format in a simple? That's really hard. How do you collapse and widen out parts of the genome you're particularly interested in? And also a pan-genome is fine one day, but a week later there could be another 100 genomes. So it's out of date really quickly. And so it's a global problem. Nobody's solved that problem yet. And I can see this community is going to be generating a lot of really valuable pan-genome data. And yet at the moment there's not really good tools to fully analyse that. That would be good generic tools that could be quickly plugged and played with additional species.
So I don't know to what extent people from this audience are already known or working with pan-genomes, but definitely that would be for many of the pathogens to create a pan-genome that would be incredibly valuable. Still, I think that for some or quite a large number of the organism groups that we are working with, or the species that we're working with, that genomes themselves still are not present. So maybe not for the viruses or for the bacteria, also there, of course, but...
Perhaps you could explain what pan-genome is, because I think it doesn't relate to my pots and pans, but...
Okay, so basically, when you've got, let's go back. As soon as you start sequencing a second strain or a third strain, then when you do the comparative analysis, you're looking for what in each of those strains have always got that gene complement, which is what you then call the core, but then you're looking at what's the variation. So what is the difference between one strain and another, and when it's only found in a single strain or is it found in a few strains, but not in the species. And so from there you can always tell if your core genome matches almost your predicted proteome, then you can say it's a very closed genome, and there isn't that much flow of information from outside. But if it's, as I saw, I think with the Rolstonia, that's got quite a small core genome, but its pan-genome is like 5 or 6 times bigger. And so that means there's a lot of flow of information coming from outside, from other strains that have yet to be sequenced, yeah. And that makes it potentially a far more variable species. And therefore, how are you going to find the emerging problematical strains in there? And what will be the genes of interest? Yeah. That's why you need effective tools to look at 100, not just 3 strains as I've illustrated here, 100 strains and then 1,000 strains globally, and then say, oh, they're regional pangenomes, and then what is the real global pangenome of that species, and what do local practices and regional differences in climate and other things and traffic and transportation actually lead to changes in the genome. And that's what I think you are grappling with, because effectively with these quarantine regulations and everything else, it's will you end up limiting a particular pangenome to a particular region of the world, yeah? If it's effective, but if it's not effective, that pangenome will slowly spread across the globe, and then you'll see the problems that arose in just one global region going right across the globe.
I believe. Okay. Yeah.
A question related to that with the pangenome. I have a feeling that the more we sequence, the more in-betweens we will find. And for legislation, you need a species definition. But will the species definition hold for the microorganisms in 10 years' time? It's maybe too big a question, but...
So I want to partly answer this. The species definition already doesn't work for plants, but we still use it in legislation. It will take the legislation decades to catch up with a change in microbe species.
I mean, what you will identify with a pangenome is you will identify hybridization zones, and to begin with, you might not even find out what the hybridization or the horizontal gene transfer species are. But eventually you will realize that in a region there must be 2 or 3 subspecies or, yeah.
But I see here 2 areas that were touched on. One is the pangenome of known pathogens, where you can have changes or shifts or difference between regions, that if they move from one region to another may cause a problem because of your varieties. And a completely other one is the unknown things we find with high-throughput sequencing, where, and Sebastian has touched on it this morning, where you need a mechanism to select which one need attention, because they're potentially a risk. And I think there are 2 different fields, but they both need some attention there, where we can, and both to the researchers and the regulators, where you can assess which one you have to look at most.
Yes, to go a bit further on this, because in fact, sometimes you do not need a lot of information to make a decision. The German authorities make a decision on grapevine Pinot Gris virus just based on the fact that in 2012, first publication of the virus, and researcher, what they do, well, behind them they have like the motto, publish or perish. You need to publish to go through your research. And that's positive, because once you put publicly a new virus, for example, people in their own country will say, hey, I have hundreds of samples in my freezer. I will just use RT-PCR to test it again. And it was a nice case on this Pinot Gris virus because 5 years or 4 years after its first discovery associated with symptoms, it has been reported in nearly all grapevine producing countries, and more often not associated, but not, I would say, not always associated with symptoms, also with symptomless samples. And based only on this distribution, the German authorities stated it was a low risk. So this element of publication is important. And we must find a way, as mentioned this morning by Adrienne, to publish as soon as possible the data, the new virus, because other researchers will say, hey, I may have a new easy-to-make publication. I am the first to find it in my continent or in my country. And in this publication, you will say it is associated with symptoms or not. And thanks to that, progressively, month after month or year after year, you can have an idea of the repartition, just because there is an incentive to publish for the researcher. So that's a point. But currently, we are blocked by the obligation of having robust data with a bit of biological characterization to make the very first publication. It triggers quite a long time sometimes. And the initiative of open plant pathology is particularly interesting because you find a virus, you just make in a repository. So people know this virus exists and can start to analyze their sample in their own countries, and you can build first information. And so that's just one point. And to go back to the question of Baldissera first, one element that is important is this QA/QC for the application of these technologies. How sure you are about the reliability of your results? And I think there are several initiatives at European or international level to try to put first guideline to set the path toward at the end, a reliable diagnostic grade techniques for plant pest analysis. That's two elements that.
Okay, so I think there are a lot of people who will find a disease in a country the first time, then they will sequence the genome and they will sit on this for a long time, this information. And this really has to stop when it's the case of diseases which are spreading, and then people are not providing the information because they're under the need to publish, so then they don't tend to put stuff out there very quickly. And there are many great examples of where people have actually released information very, very quickly. As the information is getting generated, they're putting information out there. I think one of the examples was the project I was involved in, where they did work on the wheat blast outbreak in Bangladesh. So Sophie and Kamoon at the Sainsbury Laboratory set up a website, and he put all the data as it was coming off the sequencing machine up on that website, and all blog posts about what was being done, all the analysis that was being done. That didn't prevent him from then going forward and publishing a paper. It just meant that because that was an emergency situation, that all the information that was needed was out there as quickly as possible for the community, and then in the end the paper came out a few months later. So I think we need to think about the models that we're using in emergency situations. So I can understand why somebody may decide to keep hold of a genome if it's a non-emergency situation, then they want to make their publication. But in a situation where you have a new disease and you're clear that that disease is going to spread, then you need to get that information out to people as quickly as possible so they can use it to make markers or whatever else they want to do with that data.
Makes sense, yeah?
So, and again, part of our problem on the virology side is
Try another one.
So part of our problem on the virology side is we're finding things so quickly, we can't differentiate easily between what will be the emergency and what won't. And so some of the data sharing we're talking about is actually trying to fill that baselining gap. But I think to push back to the regulators and the funders, some of that is to decide how you partition your money between the firefighting and the baselining, and decide where your priority is in terms of, there are the things that we do need to fight because they are emergency situations, but we also need to look back. So we're doing projects now on some crops that nobody's looked in the UK at that crop for 30 years, 40 years. And for the funders, I would say, start talking between the stakeholder groups, because what may be an issue for the NPPO may also be an issue for the growers group, because if you're doing surveillance, you may give good background data to the quality pathogens as well as the quarantine pathogens. So there may be some abilities to cost share.
Okay. Maybe in view of time, so we've had the meeting planned till 3:15. Maybe it would be good to try to sum up and conclude some of the things that we've seen and heard today. I don't know, Baldicera, would you be comfortable to start it off, and maybe then we could complete each other's findings a little bit, or?
Yeah. I can focus on a topic that it's in my heart and on which we are trying to work on Euphresco, which is the problem of the diversity of data, the diversity of the infrastructure. So more and more science is becoming open, and we have seen here the potential of science of data and how this data can be used to instruct diagnostic, pest risk analysis and management. There are many databases. These are databases of universities or more global databases like NCBA, QBank, just to mention a database in our field. And a problem is that the data may be there, but if you don't know all these databases, the data is hidden. And another problem is that because of the different databases, the data and the metadata are different. So from my point of view, it's very important that as a plant health community, we prioritize what kind of data are useful, and we try to harmonize the way we produce the data that are useful for our community, and that we agree on the ontology on how to describe this data to facilitate our work.
Yeah, and I think that will be definitely form the basis to go towards to more predictive type of tools and predictive risk assessments and so on. So we've also covered an immediate need to address quality assurance and quality controls in data, making data fair, as you also mentioned. I liked your remark, Diana, to rethink models of working under emergency situations, and also that we need to, so also the remark made by Adrian, that Koch's postulates, even though they are over 100 years old, that they're still as relevant today as they were back then, and which stresses the need for good metadata associated with your biological samples and genome sequences. So prioritizing the work on organisms, establishing baselines, how to prioritize, who to coordinate, and I think these are questions that as a collaborative field we need to address in the near future, and Euphresco could play an important role maybe there as well to address some of these challenges. So I don't know if anybody has something to add at this point to the meeting or to the discussions we had. And otherwise, I suggest that I hand over the floor back to Nico.
Yeah, thank you very much, Bart, for leading this whole program during the day in good order. I think what was one of the aims of this meeting was also to see what developments there are in plant health on the molecular side, what that means, but also to bring together with people that are less familiar with that but are more involved in the regulation of these pests. And we have heard some things like emerging pests, how can we prioritize? How can we prioritize also in the large amount of data we get, which is an essential element both for researchers and for regulators, because you would like to know where the highest risk is. And I think it's important, and that was illustrated today, that the researchers and the regulators go along together on this and keep on talking together on what is important for them and how to proceed. I think that's for the benefit both of the researchers, and of course some of the researchers do work already in MPPOs, so they have their colleagues, but also for researchers in research institutes. And I think also there in the research coordination via Euphresco, it's both open for MPPOs and for research institutes. MPPOs are often already involved. So I think that's one of the things I saw today, is that it's good to have this exchange and discuss the consequences, but also that we need to do that in the future to see, continue talking to each other and see what the consequences are and where the highest risks are, so that we are prepared to work on the highest risks. Anyway, I would like to thank also all the speakers again, and the audience for all the questions and the good discussions, and I wish you all a safe trip home. Thank you.